Welcome to today's lesson on the main steps in the process of genetic engineering. Today, we will explore how scientists manipulate an organism's genes to introduce new traits or enhance existing ones. In today's lesson, we will cover the specific stages involved in the genetic engineering process: isolation, insertion, and introduction. Our goal is to ensure you can describe each of these steps clearly and understand their importance. So far, you have learned about what genetic engineering is. You understand that it involves modifying an organism's DNA to achieve desired traits, which sets the foundation for our deeper dive into the process itself. Understanding the steps of genetic engineering is a crucial advancement in our biological understanding. It allows us to comprehend how scientists can create genetically modified organisms, develop new medical treatments, and solve various biological challenges. Let's get started. Let's dive into the fascinating world of genetic engineering, a process that allows scientists to modify the DNA of living organisms to introduce new traits or capabilities. The main steps in genetic engineering can be broken down into three key stages: isolation, insertion, and introduction. The first step in genetic engineering is to isolate the gene that scientists want to insert into another organism. This gene might be responsible for a useful trait, such as disease resistance in plants or the production of human insulin in bacteria. To isolate this gene, scientists use special enzymes called restriction enzymes. These enzymes act like molecular scissors, cutting the DNA at specific sequences to extract the desired gene. The key takeaway from this is isolation involves using restriction enzymes to cut out a specific gene from an organism's DNA. Once the desired gene is isolated, the next step is to insert it into a vector. A vector is a DNA molecule used as a vehicle to transfer the target gene into the recipient organism. Two common types of vectors are bacterial plasmids and viruses. Plasmids are small, circular DNA molecules found in bacteria that can replicate independently of chromosomal DNA. Some viruses can be engineered to carry the desired gene and introduce it into the host organism's cells. To insert the gene into the vector, scientists again use enzymes to cut the vector DNA and join the gene that is being inserted into the vector. The key takeaway from this is that insertion involves using vectors, such as bacterial plasmids or viruses, to carry and transfer the isolated gene. In the final step, the vector containing the desired gene is introduced into the target organism's cells. This process varies depending on the type of organism into which the gene is being inserted. The gene is transferred to the cells of animals, plants, or microorganisms at an early stage in their development so that they develop with desired characteristics. The main goal is for the gene to integrate into the target organism's genome and be expressed, thereby endowing the organism with new traits. The key takeaway from this is that introduction involves delivering the vector into the target organism's cells, allowing the new gene to be integrated and expressed. Before we finish, let's review the history of a very important genetically engineered organism. One of the most surprising developments in genetic engineering, first developed by Boyer and Cohen in 1972, was the creation of the first transgenic animal. In 1982, a mouse with a foreign gene inserted into its genome, known as the oncomouse, was engineered to develop cancer for research purposes. This breakthrough marked a significant milestone, demonstrating the potential to modify complex organisms, such as mammals, beyond bacteria and plants. In today's lesson, we further explored genetic engineering. This included a more detailed exploration of the main steps involved in the genetic engineering process. First, we identified the gene of interest from the donor organism. This gene is responsible for the desired trait that we aim to introduce into a different organism. Next, we discussed the isolation of this gene using restriction enzymes, which act like molecular scissors to cut DNA at specific sequences. Following the isolation, we need to insert the gene into a suitable vector, typically a plasmid or virus. We covered the process of using enzymes to incorporate this DNA into the vector for insertion. After the vector is prepared, we then introduce it into a host organism. This allows the host to express the new gene, producing the desired protein or trait. These genes are often transferred into the host at an early stage so that they can develop with the desired characteristics. In summary, you should now have a clear understanding of how genetic engineering is carried out, from identifying and isolating a gene, to inserting it into a vector and transforming a host organism.